Welcome again to the statistical seminar. Today, it's my pleasure to introduce Dr. Suprateek Kundu from Emory University. He is assistant professor from the Department of Biostatistics and the Bioinformatics. His research is focused on developing practical statistical methods and theory motivated by high-dimensional biomedical application in epidemiology, neuroimaging, and multi-omics data. So methodologically, he's interested in developing graphical models and variable selection. In terms of application, he's interested in inferring brain networks using fMRI data as a function of clinical, demographic, and genetic information, inferring dynamic brain networks which change over time, structure connectivity problems, entailing analysis of complex diffusion, MRI data, and integrative approach for imaging genetics involving multimodal data. So Dr. Kundu, if I can ask you to share your slides and
yeah, yes, absolutely. And thanks for the wonderful introduction. Can you all hear me?
Yes.
Okay, great. So I'm just gonna do this in a full screen mode, and hopefully you can see my screen.
Well, right now it's still it's not a full screen mode for me.
Yeah. I guess this is working. Yeah. So
yeah, it's okay. Yeah. Sorry.
Good. Yeah. So thank you for the invitation, Yang, and I'm really excited to talk about some of my ongoing research. And it's always nice to come back to Texas A&M. I used to be a postdoc here several years back, so it's always good to come back. So today I'm going to talk about some of my work on integrative learning and joint learning. And in particular, I'm going to focus on joint learning of multiple high-dimensional scalar-on-image regressions. I'm going to motivate the data that I'm using, which is brain imaging. And I'm going to motivate the need for joint learning. I'm also going to talk about a few previous works that we have done on joint learning. And then the main chunk of today's talk is going to be an ongoing work. It's not complete by any means. It's still work in progress. But I'm looking for feedback. So I'll get started. This is a joint work with my doctoral student, Shin Ma. And this work is supported by NIH funding, which I've acknowledged at the end. So today's talk, again, is concerning brain imaging data. So I want to start off with MRI data. So MRI is a major component of neuroimaging studies, and it illustrates the brain anatomy and brain activity in a safe, non-invasive way. So the good thing about MRIs and fMRIs is you don't have to slice open the head so you can just do it in a non-invasive way, and you can measure brain anatomy and brain activity using these kind of images. So there are two kinds of images that I'm going to talk about today. One is the structural or the T1 weighted MRI images, and the other one is the functional MRI images. So the T1 weighted MRI images provide information about the volume and so on, the brain anatomy, yeah, and it's used heavily in medical imaging. And functional images, of course, measure the blood oxygenation levels in the brain, and it stands as a proxy or approximation for the brain activity. So functional MRI measures brain activity, whereas T1 weighted images measure the brain anatomy or the brain structure. So brain images are intrinsically high dimensional. So there are it's a 3D image. And for functional data MRI, you have a 3D brain image, and then you collect that repeatedly in a sequence. So you have a time series of 3D images. So it's actually 4D data. So the functional fMRI data looks like a time series, but instead of having one time series, you have a collection of multivariate vector of time series, and the vector corresponds to different voxels in the brain, and these voxels are spatially correlated. So what are the experimental setups of interest, and where does the joint learning and the joint modeling come into the picture? So our setting of interest includes studies where multiple brain images or features are available on the same set of individuals. So for example, you could have collected task and rest fMRI data on the same individual, and you have a group of individuals. So you would like to ideally leverage the common patterns of information in the brain when you're looking at task and rest. Or you could even have longitudinal brain images collected at baseline and follow-up visits. So you could have longitudinal images, and you would like, for example, in Alzheimer's, in the acne data set, you have Alzheimer's data, which is collected over time, and you would like to kind of share or leverage information across visits. So there could be many, many different applications of this kind of a joint modeling. The first figure here is something called a Stroop task, which is at the end of the day a vision task, and this is fMRI data. And so you can see that the data was collected from the same individual when they were relaxed and they were doing the task in exertion, right? So and then we had a group of individuals. And then of course the second figure is the MMSE score. MMSE score is a cognitive measurement, and this is from the acne study. So you can see that there are three groups of scores: the Alzheimer's, the MCI, and the cognitively normal. So you can see that over time, the x-axis represents time, and over time, the cognition score of Alzheimer's really starts to come down, whereas for cognitively normal, it stays the same. So there's clearly this longitudinal progression, and and there is a need to borrow information across these time points. So this need for joint modeling really stems from a growing appreciation of data fusion approaches in medical imaging. Yeah, so there's a growing emphasis on data fusion in medical images, but there are challenges that still exist. So for example, the brain images are noisy, and there are pre-processing steps that one can use, for example, motion correction, to remove some of the noise. But there are other sources of noise, for example, in the scanner, that are difficult to handle and that just cannot be filtered out. So the noise in the fMRI data is known to sometimes have a low signal to noise ratio. So the noisy images can reduce the signal strength and power. And really the hope is that by doing a joint analysis of multiple related data sets, it'll provide us with a promising avenue to boost signal and mitigate the influence of the noise. And of course, another big thing in neuroimaging and neuroscience and in fMRI and MRI now is reproducible findings. It's a big emphasis by the NIH. And so hopefully by doing joint analysis, our findings are expected to be more reproducible. However, the literature on statistical methods for integrative analysis of multiple brain images or multiple images is somewhat still sparse. So in a recent work, we proposed an approach called Bayesian joint modeling for multiple brain networks. So really what this does is it helps us model multiple brain networks jointly. And the way it works is by kind of simultaneously pooling information for the edges. So there are certain latent components which are shared between the data sets, and there are certain unique components that are unique to each data set. And I'm going to talk a little more about this in a minute. And then we took this approach and then we modified it to longitudinal acne data. And we used this approach to analyze, to compute networks, brain networks for longitudinal acne data by sharing information across the visits. So this has kind of, in a really nice method, has a lot of implications for joint learning of multiple networks. And the way this method works really is, so suppose there are two data sets or two, Y1 and Y2, these are fMRI data sets, and we use a Gaussian graphical model to calculate omega-1 and omega-2, which are sparse precision matrices. And omega-1 and omega-2 have certain, the probability, right? So there's an edge with a high probability or a low probability. And the way we do this is by modeling the probability with a link function h, and then having certain embedded latent factors eta naught and eta 1 in this so h could be, for example, a logit link or a probit link, right? it's a parametric link function, and eta naught is the shared component that is shared by multiple data sets, for example, rest and task, or between visits. And eta 1 and eta 2 are unique or differential components, latent factors that lead to differences between data sets. So, and then we impose a Dirichlet process prior on these etas, and we get a really a flexible model on the edge probability. So the underlying mechanism of this approach is in sharing latent components, latent factors over data sets. Okay, so that is all about joint learning of multiple networks. So just to emphasize the application a little bit more in the beginning, our motivating example for today's talk, which is again a work in progress, is acne data. And so the acne data is Alzheimer's data, and some of you are already familiar with this kind of a data set. And so it has information, longitudinal neuroimaging, clinical behavioral information at baseline, 6 months, 12 months, and so on for cognitively normal, mild cognitive impairment, and Alzheimer's disease, right? So so it's well known that Alzheimer's disease is a neurodegenerative disease, and it progresses over time. And the outcome that we want to model is a cognitive measure. So for our applications, it could be the MMSE score. The predictor is the 3D structural MRI. So in the previous two papers that I talked about, the brain images corresponded to functional MRI. But here, for this talk, it's going to be the structural MRI data. And the way we want to do this is use a Scalar-on-Image regression model to predict the outcome of interest using the brain image. However, we are going a step further and saying, instead of just building one Scalar-on-Image regression model, can we jointly model multiple Scalar-on-Image regression model, and each model corresponds to the relationship between the outcome and the image at a given longitudinal visit. Okay, and again, this method is not restricted for longitudinal data. It could be multiple related data sets, right? And then we, of course, we do this analysis separately for each disease group. Yeah. So again, the goal here is to borrow common patterns and leverage patterns of association across visits. So again, we have had some success in our previous joint network modeling work using brain images, but there are several things and aspects that need more investigation. And so for this work, we're interested in joint predictive modeling for multiple high-dimensional Scalar-on-Image regressions. So, but it is not immediately clear how to construct these joint predictive models that are able to leverage commonness patterns across multiple data sets, while respecting the inherent heterogeneity and differences. Another key question for us is how to tackle the noisy brain images, right, under the joint modeling framework. How do we tackle the noise in the data? And what kind of theoretical guarantees can we expect if we were to do this? So our proposal is a novel approach for joint analysis of multiple Scalar-on-Image regression models. So the idea is to first use a dimension reduction. So let's say wavelet decomposition to represent the MRI images. So the nice thing about wavelets is they retain the spatial information, and then model the data set specific wavelet coefficients using appropriate penalties to pool information across multiple data sources. So we can use penalties on the wavelet coefficients to suitably penalize across and borrow information across multiple data sources and smooth across multiple data sources. We propose two estimators. One is, and so this is going to be a frequentist penalized approach, or M estimator, right? So then one is based on a group bridge penalty. The group bridge penalty has a hierarchical representation. It has a nice interpretability feature. So we first investigated this. And of course, a natural candidate for these kind of settings is also the group lasso penalty. So we would also like to investigate the group lasso and the properties of this estimator. So, and we would of course like to develop an efficient computational algorithm for M estimation for noiseless images, so assuming there is no noise, and then extend this approach for cases when there is actually some noise in the data. So, but before we proceed, it's always important to give some context and where the literature stands. And of course, there is a huge literature on functional data analysis, which is beyond the scope of this talk. Specifically for scalar-on-image regression, there has been some work in the last 7, 8, 10 years. And of course, there is some work on Bayesian voxels-wise regression methods. And these approaches impose suitable priors for sparsity and smoothness. And then there is a tensor regression approach. And then moving on to more penalized approaches, there are tensor regression approaches. And there are approaches which use total variation norm. And more recently, a paper from Yale, which uses an internal variation norm for a 3D brain image. In general, most of these approaches are focused on 2D images, but really in brain images at its original sense, 3D. Yeah. So this recent paper addresses a 3D brain images. And there has been work on representing the brain images using wavelets, which are computationally efficient. And there's a paper in Annals of Applied Statistics by Wang et al. 2014. Of course, there are other approaches that use images in functional linear models. However, there are no mainstream proposals for joint modeling, to our knowledge, for multiple scalar-on-image regression images. And of course, the issue of noise has not been thoroughly investigated as far as we understand. So, we propose a group bridge or a GB penalty motivated by a hierarchical representation and theoretical justifications. And what we do is we establish non-asymptotic error bounds under the group bridge penalty. And we note that the theoretical properties of the group lasso penalty are also well established in literature, which will carry over to our case. So this is for the noiseless setup as of now. And then for noisy images, we generalize our method to settings such that with settings with an additive error. So the error is additive, and then we come in, and this setting involves P being the number of voxels in the image. It increases at a rate faster than N. And computationally, for this case, we propose a composite gradient descent algorithm for computing the M estimators that can be computed in polynomial time. And under certain commonly assumed regularity conditions, we are able to show non-asymptotic error bounds for both the penalties. And we also show that the projected gradient descent algorithm is guaranteed to converge at a geometric rate to the solution of the original problem. So in essence, although the gradient descent algorithm is a kind of an approximation of the true solution, it converges to the true solution pretty fast. And so these are the kind of things that we want to do, but how do we go about doing such a thing? And so before we dive into the notations, kind of looking at the big picture, which is the significance and innovation of this work, which is methodologically, this is one of the first approaches for joint estimation for multiple scalar-on-image regressions. And we are, to our knowledge, one of the first to establish non-asymptotic statistical error bounds with noisy images for P greater than N settings under both the non-convex group bridge penalty and the group lasso penalties, and propose some bounds also on the optimization error. Of course, there is past theoretical work on M estimators under noisy images that have focused on P less than N settings, and some recent work on P greater than N settings. However, these approaches typically use a L1 penalty and cannot be straightforward in a straightforward way extended to joint modeling for multiple images. And one last thing is there's a tricky part about defining these models for noisy images, which is these methods or the methods out there in literature require some kind of an estimate or knowledge about the noise covariance. So these methods will, will require a term which is the noise covariance with under an additive error. And for our data, which is the acne data, what we do is we have longitudinal images from stable, healthy controls. These subjects do not show any disease progression. They're healthy, and we have data for them at baseline and over multiple visits. And so we use the scans from these subjects to calculate this noise covariance. So the assumption is that any variation in the images of these individuals who are perfectly healthy and have no progression, they're stable, any variation for the brains of these individuals are representative of the noise. And so we calculate the noise covariance using this data, and we use this same noise covariance for the Alzheimer's and the MCI group. Of course, the Alzheimer's group people have variation in the brain image across time, which is due to progression of the disease, but that is separate from the variation due to the noise as well. Okay. So some notations. We have M data sources, M equal to 1 to capital M. M data sources, NM samples. YMI is the scalar response for the M data source. And the ith individual. XMI is the functional predictor. So U is the voxel coordinate. So U can be 2D or 3D. We work with 3D. It also can be 2D, doesn't really matter. And beta MU is the functional regression coefficient. So this is the coefficient that captures the relationship between the scalar response and the functional predictor. So this is a standard functional linear model setup. And so under a wavelet decomposition, the model starts to look like a linear model, C being the wavelet coefficients, and eta being the wavelet coefficients, and C are coefficients that come from the image. So these can be directly calculated. And so what we do here, we propose a hierarchical structure for the wavelet coefficients. So eta mj, which is the jth wavelet coefficient for the mth data source, we say, okay, let us describe this using a hierarchical structure. So break this up into a component gj, which is common across the data sources. It really doesn't change. And then a zeta mj, which is data source specific, right? So this has a kind of a nice hierarchical structure, and it's interpretable. But these components themselves may not be identifiable, but when they're zero, we can identify those. But mainly we are interested in this guy, which is eta. So the hope is that this component GJ will, we will learn this by pooling information across the images, which will lead to a joint estimation. So what kind of penalties do we put on these components? So we impose separate L1 penalties on G and zeta, and then we can show that this actually reduces to a more simple term with just one penalty lambda, which is a function of these two original penalties. And the estimates here can be computed in an iterative manner using the lasso, right? So using the lasso, we can get G conditional on zeta, and then conditional on G, we can get zeta using the lasso. And so this equality follows from lemma 1 in the paper. And then of course, it is also possible to show that the solution of eta obtained by this optimization is equal to at least one solution obtained by this group bridge penalty. So if you solve for eta and for this solution and get in value, then and then if you solve for eta using this group bridge, you will get a solution which will look exactly the same as your solution here. Of course, this is a non-convex problem because the group bridge penalty is non-convex. And so the computation, how does it proceed? Well, we already showed that it can proceed via an iterative lasso, and it is guaranteed that the solution will reach a local maximum, local optimum in using via lemma in the paper. And we have a tuning parameter lambda, which is the penalty term. And of course, there's always a question of what kind of wavelets to choose. There are several different choices out there using the Haar wavelets and so on. And depending on the image, this is a choice that any wavelet method needs to decide in the first. That's a standard issue in a wavelet method. Yeah. So this is a result which shows that the group bridge has a weak oracle property. So under certain reasonable regularity conditions, then for large sample size with high probability greater than this term, with high probability, there exists a local maximizer for eta such that the true zeros in the eta will be identified, and for the non-zero etas, the estimate will converge to those true values in some sense, okay? And S here is the sparsity level in the true model. So again, what this result means is that when eta is truly zero, then the estimate will also be zero for large N with extremely high probability. And for when the eta is not zero, then it will gradually concentrate in increasingly small neighborhoods of the true eta norm. Okay, and of course, these bounds will vanish to zero as N goes to infinity, but this is again kind of a finite sample bound. And so, of course, using this result, it is then possible to show that the original regression coefficient beta, remember these etas are the wavelet coefficients. So the original regression coefficients beta are also converged to the true beta. And that's a direct outcome of this result. So, of course, then we are also interested in exploring the group lasso. So the group lasso penalty obviously looks something similar to this, and there are well-defined properties of the group lasso. Of course, when a group lasso is able to consistently estimate the true parameter and so on, and all these properties will transfer over to our case as well. Of course, the key difference between the group lasso and the group bridge is the group bridge is non-convex, whereas the group lasso is convex. Okay. So this is, up to this point, we've just talked about the noiseless case. And a couple of other things I don't want to get into for the talk. I mean, the group bridge has a stronger recovery for the sparsity patterns compared to the group lasso, but that's those kind of things. We can talk about that afterwards, perhaps. So now, how do we extend this method for noisy images? So the goal is, okay, we have a method which works, which is a kind of extension of functional linear model type approaches. But now, what happens if the wavelets are noisy? Of course, if the images are noisy. If the images are noisy, then these methods which do not account for noise obviously are not going to do well. And so for our purposes, we assume an additive noise model. So at each voxel U, we assume that the observed image is X tilde, which looks like this, W transpose theta. And then this true image is actually X, which is C transpose theta. And so W is equal to C plus U. U is a zero mean sub-Gaussian random variable. Yeah. And this is actually, this corresponds to the true image, but in reality, we observe the image X tilde, which is W transpose theta, where W is now corrupted by this noise U. So U is a zero mean sub-Gaussian variable, and C can be fixed or it can also be sub-Gaussian as well. And so assuming the error variance on U, sigma UU is known, okay, and the theory also extends to the case when sigma U is not known, but can be estimated with repeated images. but for the purposes of today, let's assume sigma UU is known. And so then this is the modified M estimator that is we propose. And so what this does, it kind of this, if you look at this, this looks similar to what you would get from a, let's say, lasso. If this was a lasso penalty, this looks like a lasso, but now this gamma hat is really the adjusted X transpose X matrix, so to speak. And the lowercase gamma hat is W transpose Y. Remember, W represents the coefficients coming from the corrupted image. And really then what happens, and so this gamma hat, because of the noise, this is not at all guaranteed to be positive definite. In fact, it is not positive semi-definite, and it could even have negative eigenvalues. And if this has negative eigenvalues, this whole thing tends to start to blow up, okay? And then, so to tackle this kind of a situation, we need some kind of a trick. And the trick is we keep the solution to a certain ball, restricted to a certain subspace or a ball. And the hope is that by restricting the solution to this kind of a ball, we will be able to get a meaningful solution, right? And so we try to see what happens if we solve for this kind of a solution when rho is a group bridge or a group lasso penalty. And the motivation for this kind of solution comes from a collection of papers by Wainwright and colleagues a few years back. Yeah. So, but they only explored L1 penalties and so on. But our case is general, and involving other types of penalties which involve a grouping behavior and which involve images and so on. And so what we show here, that under an additive measurement model, provided the true eta naught is k sparse, which means that the number of non-zero coefficients at most k, and the true eta naught has a certain, the norm is bounded, then for sufficiently large penalty parameter and sample size of n greater than k times log p, with a probability at least this, for both the group bridge and the group lasso penalty, we are able to find an upper bound for the statistical error. So you can see that eta hat minus eta naught is actually the statistical error. Eta hat is a solution to our regularized problem. Eta naught is the true coefficient. So the statistical error is bounded. And under the assumptions of the theorem, this error goes to, and this is again a non-asymptotic bound, and this error will go to 0 as long as n is greater than k times log p. Okay, so the error is measured both in terms of L2 norm and as well as the L1 norm. Okay. So now the question is, yes, going back to this criteria, we have defined this modified M estimator, but how do we really calculate a solution for this M estimator? If you were to directly try to find a solution here, it's not that straightforward. So then for this, computationally to find a solution, we propose a projected gradient descent algorithm, which iterates along with these steps. And it can be shown that this update can be done using a projection. So a projection onto the L2 ball, of the L2 projection onto the ball, which looks like this. So this ball right here. So and this solution can be obtained rapidly in OP time using a procedure due to Dushi in 2012. Okay, so the bottom line, although the original solution for the original problem can be hard to obtain, one can do a kind of a projected gradient descent algorithm and get a solution which is approximate. It's not exact, but it's computationally friendly. It can be obtained fast, and it's kind of an approximation to the original solution. But now the question is, how good is this approximation? Okay. And so the next result shows us that for the group lasso penalty, we found that the result was very hard to derive for the group bridge. But for the group lasso penalty, the optimization error, so this is the optimization error. So this is the error between the iterate eta t from the projected gradient descent and eta hat, which is the solution of the original problem. This guy is bounded by the statistical error. So remember, the statistical error is eta hat minus eta naught. So this is the statistical error. So for large enough, for all iterations t greater than capital T, and this capital T can also be characterized. So if t is large enough, then the optimization error is bounded by the statistical error. And we have already shown in the previous theorem here that the statistical error is already bounded, right? So really, what this result shows us is that the optimization error under group lasso is essentially bounded by the statistical error. And this result guarantees us that the projected gradient descent produces an output that is essentially as good in terms of statistical error as the optimum of the original program. So broadly, that is what the methods and the like. I think in the interest of time, I'll move on to simulations. We did some simulation studies. Now, the results I have today on the slides just correspond to the simulations that are without noise. We have results for noisy, the noise part, and I'm going to touch upon that. But the next few slides are results for the noiseless part. So we implemented our method, and then we tried different wavelets, the Haar wavelet and the Debussy wavelet, and we compared with the WNet and a couple of other procedures. And the last one really is the lasso, which is an independent or separate estimation of these data sets, not a joint estimation. And we look at out of sample prediction MSE, and we look at the bias in recovering the true coefficients. So we did 3 images, and they have certain shapes. So the shapes of these, of course, these 3 images in each setup are 3 related images, and their shapes are similar, but the sizes are different and the positioning is slightly different. And the same goes for these 3 images. You can see that the shape, the size of the area is much smaller for the 3rd image. So we tried these fuzzy round sharp, square sharp, and triangle sharp. So we kind of looked at these 3 images. Remember, these are 4 different scenarios, and for each scenario, there are 3 data sets. So this is the signal. This is not the image actually, but this is the signal, the coefficient map, the true eta naught, the coefficient map. And our data sources have sample size 100, 200, and 200. And the image size is 64 by 64. So again, this is a 2D result. And the reason I included this, it's easier to visualize. 3D is slightly harder to visualize. We also have 3D results that I'm going to touch on. This is what we found. So for the first 2 images, we found that, so this is our method, the proposed method using the group bridge, and this is using the group lasso. So you can see it kind of starts to reasonably recover the true coefficient signals. And this is a competing method, and this is the separate estimation of the 3 data sets. And you can see completely the results kind of, the signal is not, it's very hard to recover the true signal if you do separate estimation, and so on. And then the other 2 shapes look like this. Again, the first, the second and the third row is the group bridge and the group lasso. And it's reasonably recovering the true signal. Of course, there are it could do much better in some places. And these are these two competing approaches. Okay, and so what about the prediction MSE? So this is these two are the group bridge method, but with two different wavelets. And this is the group lasso with these two different wavelets. Yeah, so these are the group 1, group 2, group 3, and these are the different shapes that I talked about. Okay, and what we find here is that if these bolded figures represent significantly improved prediction. Right? So it's of course we can do many more simulations, but broadly what we found is captured by this slide here, which is if you use the joint estimation method, you stand to gain, and you get a reduction in the MSE across the board. And this is a corresponding slide which calculates the bias. So the bias in recovering the true coefficients, and it is evident that the group bridge does better. Now this is the case where this is the noiseless scenario, right? So the group bridge tends to do better. And before we move on to the real data, for the interest of time, I could not include slides for the 3D images. So 3D also, we found similar results. Of course, the thing with 3D is the computation is a little more burdensome, but it's still manageable. Yeah, so we used a 64 by 64 by 64. And we found more or less similar results. So let's talk about the longitudinal acne data analysis, right? So we have the longitudinal acne data involving MRI images. And so I've already talked about this at the beginning a little bit. And so we measured the cognitive scores and other demographic and clinical variables. And we use 1.5T MRI images. And so in acne, there are 1.5T and 3T images. The 3T images are kind of state of the art and higher resolution, but the sample size is higher for 1.5T in acne 1. acne 2 and acne 3, acne Go, they have mostly 3D images, but we are using acne 1 images. So we are going to look at the prediction results for 80 individuals only for this talk. So for 80 individuals, there were 188 individuals at baseline, 133 individuals at 6 months, and 135 individuals at 12-month visit. And so our analysis requires data on the same individual across all visits or all data sources. So we stick with this 135 80 individuals who had data on all of these visits. Of course, please note that acne has extended follow-ups also. So there is a 24-month and so on, right? And so we also included covariates such as age, sex, APOE status, the intracranial volume at each visit, and so on. And so what we find here, again, this is the noiseless version of the method. And we find that the group bridge with the Debaucher wavelet and the Haar wavelet and the group lasso with the same two wavelets, the group lasso tends to do best actually in acne for the noiseless setting. And these last three panels are this is again this is a joint estimation approach also at the WNet, but it's slightly higher prediction, and the last box plot is the lasso, which does a separate estimation, right? So this is a prediction MSE. So as I mentioned in the very beginning, this is a work in progress, and so we also have numerical simulations for 3D, as I was mentioning, which I did not present today, and we did also do the projected gradient descent algorithm for noisy images via simulation studies, and we found that the group bridge and the group lasso penalties for the methods for the noisy images are have good results when the sigma UU, the noise covariance matrix, is assumed to be known. If the noise covariance matrix is completely unknown and there's no good estimator for it, then this method and most of these other noisy covariate approaches might not do that well. And so currently we are actually implementing the noisy covariate scenario and algorithm for the acne analysis. So that's currently ongoing. We're applying it to the AD and the MCI cohorts. And the good thing is we can estimate the noise covariance in the acne data using the imaging data from healthy controls, the longitudinal imaging data from stable healthy controls. So that's another added appealing advantage of this data set and methodology. Okay, so in summary, we develop a novel approach for the joint analysis of multiple high-dimensional scalar-on-image regression models, and we develop efficient computation for these models. And the thing is, because this is 3D data and spatially distributed voxels, it's really very appealing to do a Bayesian approach, which it's worth thinking about it. But of course, as a first pass, this is just an approach involving M estimation, because with the Bayesian approach, of course, the computational intensity increases very quickly. And so then this work, we believe, is one of our first attempts to develop a joint estimation for multiple scalar-on-image regression models involving noisy images. Again, this is a couple of tweaks are needed to kind of get final few things in place, and we hope to have a completed version of the manuscript later on in the fall. I would like to thank the NIH for their and the audience for sticking through the talk, and the organizers once again. And at this point, I'm happy to take any questions.
Okay, great. Very nice talk. Do we have any question for Suprateek?
Yes.
Okay, I may have one. Suprateek, thank you very much for nice talk. It was very informative, and I'm glad that you use wavelets. I may have some wavelet advices for maybe improving things.
Yeah.
One thing is that you, for conditions of your theorem that you showed convergence rates, the conditions depend on wavelets, because you have this K sparse situation among the coefficients, and also the norms of the coefficients are bounded. That really depends on, for the same image, that depends on the wavelet. So in your situation, the selection of wavelet, which is usually not important, is very important. In this case, wavelets, they correlate. So you, with bad selection of wavelet, you may lose the correlation structure in the 3D image. So maybe instead of going with Haar or Dalbesh's, one thing to consider would be to have these non-separable wavelets that are mindful of the correlation structure.
Uh-huh.
And maybe since you have predictive modeling, once you estimate your coefficients, you may go back with these non-separable models and get prediction.
Yeah.
So
yeah.
My point is that in this case, the choice of wavelets, which usually does not matter in smoothing, denoising, is important.
Yeah, that's an excellent point, and I couldn't agree more. I think in imaging applications, especially with 3D images, it's very important which wavelets are chosen, and there are no golden principles or guidelines to actually select these wavelets. So I think, your point is well taken.
way to select is maybe to look at the images and make the data generated wavelets. This is possible, but in 3D case, I don't think it is done. In 2D, 1D is done.
Okay.
So maybe this is something to think about.
Like an empirical approach.
Empirical approach to select further wavelets. In
Yeah.
this case, maybe not separable, but this is something to link with signal processing people.
Right.
See how to do it.
Yeah. Okay. Yeah. Certainly. Yeah. That's a great suggestion. Thank you.
Wu, do you have a question?
Yeah. Hello, Dr. Kundu. I found your talk very interesting, and I have a questions on the simulation setup. And yeah, so in your simulation, I found that your data set has like some similarities on the shape of the signals, right? So some of them have a square shaped signals, and some of them has a triangular shaped signal. So I'm just curious if we fit the proposed model on the data set, which have a different shaped signal. So example, one group has a square shaped signal and like another group has like a triangular shaped signal, then like the model will gonna fit the similarities, or it will gonna fail to capture the similarities.
Yeah, no, thanks for that question. So that's actually a very pertinent question. So it is entirely possible that the shapes are different across the images. And we did try that, and we found that if the shapes are vastly different, then our approach and most joint learning approaches are going to fail. Having said that, for our applications involving longitudinal images, we don't expect the signal in the brain to drastically change in shape, because that would mean that completely different configurations of voxels are getting activated over different visits, which is not a biological reality. So for our particular application, yes, we do expect these shapes to be somewhat retained over visits, but of course, there could be exceptions, and there could be other applications where the shape is completely different across the images, in which case I have to say that most joint estimation methods may not do that well in my experience. Yes. Thank you.
Thank you so much.
Any other questions for the speaker? Val, please, yes. You are muted.
So very nice talk. So I guess more from a substantive point of view, have you analyzed these images, and has the analysis led to any insights into the structures in the brain that are related to any of these diseases? And are the results at all interpretable, I guess, biologically?
Yeah, that's always the question with these kind of data sets. Yes, so we found very strong activations in certain parts of the hippocampus when predicting the cognitive score for Alzheimer's, and it is known that the hippocampus indeed shrinks in people who have Alzheimer's. So we did find strong activations in the hippocampus area, which is validated by existing studies as well. And of course, the analysis that I presented today for acne, it's not the whole brain. It's a 64 by 64 by 64 cube comprising of the two hippocampi. And we found very strong signals within the left and the right hippocampus, but feeble signals outside of the hippocampus mask, which leads us to believe that these activations are biologically interpretable. We do not have any novel activation insights as of now, but I'm looking forward to the noisy case, because I feel like the fMRI data has noise, the MRI data has noise, and the noisy image analysis might actually give us some exciting insights in the data set.
So I guess the MRI images are typically more on order of 512 by 512 by 256 or something like that. So yeah, I mean, can you apply the algorithms to the full MR image sets? Or,
yeah. Yeah, so that's the goal. Definitely, we haven't tried that. We haven't tried feeding the full 3D brain image into the model, and we were actually a little apprehensive to do that. And most of these Scalar-on-Image regression literature that is out there hasn't done that kind of a thing. I think the most recent paper by Heping Zhang just came out in 2020 tries to do that using an internal variation penalty. But the traditional and overwhelming majority of articles, they have taken sections of the brain and tried to do the prediction. And these selected hippocampus and other sections make biological sense. So they're selected not randomly, but using expert domain knowledge. Because if you look at disease like Alzheimer's, there are large chunks of the brain that are not activated, that are not related to the disease, but there are other certain parts of the brain, such as the hippocampus, that are very strongly related to the disease.
Thanks.
Yeah, but that's a great idea. I think it's definitely worth a shot, at least from a computational standpoint, to see if we can push through this high volume 3D image into the model and what happens.
Well, I have a quick question. So in your model, you really treat the groups sort of exchangeable. So but in your application is actually as a longitudinal data. So the groups is actually the visits. So you have baseline, 6 months, 12 months. And you also mentioned that the acne, the study was actually collecting more and more, time points. So I guess maybe it's a reasonable thing to extend your current model by incorporating the ordering or partial, conditional independence of the groups if you have this sort of longitudinal data. So I'm just wondering if it's possible to extend your current methodology to allow that conditional independence of the groups.
Yeah, absolutely. So we looked at, just for today's results, we looked at one acne group over time. And so there is no ordering built into the model, as you rightly said. It will certainly be very, very exciting to build certain kind of ordering into the related databases, maybe using a spline or some kind of a monotonic trend function. It is tricky though, because there are large chunks of the brain which will not show any activation for all visits, whereas there are other parts of the brain which might show an increasing or decreasing trend. I'm not sure if that answers your question, but yes, it will be certainly exciting to build some kind of a ordering into these multiple data sets, with a caveat to recognize that most chunks of the parts of the brain will not have that ordering, will just be flat or zero throughout.
Thanks. Any other questions for the speaker? Okay, if there's no other question, let's thank our speaker. Yeah? Very nice talk. Thanks.
Thank you.
Thank you.